Yeah, thanks a lot, Matthew, for the invitation, and to everyone for joining us today. I hope you find the talk interesting. So I will start sharing my screen. Can you see the presentation?
Yep, that's showing up.
Okay, perfect, great. So hi everyone, thanks a lot for joining us today. I will talk to you about today about the work, a side project I was doing during my PhD. That was started by my PhD advisor, Jose Crispin in Boston, and then we finished in Mexico. So that's why probably you see all these affiliations down here, just to make sure it was a traveling project. So Samantha and others before in the previous talks, they have made quite clear that the immune system needs to be under homeostasis, because it's going to protect us against infection and, for example, microbes, but also tumors. But also you need to control the activity of the immune system, because otherwise you can develop autoimmune diseases or allergies. So therefore this example of the yin and yang of the balance. And if you get too much suppression of the immune system, then you're susceptible to infection and also tumor development. But if you have a very excited immune system, then people that can predispose to autoimmune disease. Something about autoimmune diseases is that usually they are not monogenic. So some autoimmune diseases are due to alterations in one gene and one protein, but many of them are multifactorial. So you have a genetic component and also an environmental component. And the genetic component usually is not due to just one gene, but several, the influence of several genes. So my talk today is going to be about CD8. So briefly, for those that maybe are not that familiar with T cell activation and how they respond to pathogens. So for example, we have a pathogen like a virus that is going to be our antigen for the T cells. The molecule they are going to recognize eventually. And dendritic cells that, again, we got amazing talks from previous speakers, are going to present these antigens to T cells that then are going to get activated and start proliferating to counteract and destroy the pathogen. So as you can see here, in general, when you have an infection, a pathogen, a virus in this case, for example, the virus or the bacteria is going to expand. And then the T cells, first the innate immune system is going to act, trying to control this infection. But eventually the T cells are going to be activated and they are going to expand, as you can see here, to try to clear the infection. But then in order for the immune system to go back to homeostasis, because otherwise you will have chronic inflammation, and that's not also, that's detrimental for us, you will have a contraction phase where the T cells will go back to basal levels, or at least decrease quite a bit in numbers. And then they will form what is called the memory T cell that we are familiar and we are trying to look to generate now with all these vaccines, for example, for coronavirus. It's important to note that the contraction phase needs to happen, otherwise, again, you have chronic inflammation. And classically, there's two mechanisms that induce the contraction or the death of these viral specific T cells, and this is what is called reactivation-induced cell death and cytokine withdrawal-induced cell death. And classically, as you can see, the reactivation-induced cell death will happen at the peak of the response of the T cells, while the cytokine withdrawal-induced death happen a little bit later on. And classically, they have been attributed to the activity of two molecules that act on T cells that are FAS and BIM. However, as you can see here from the red and the blue dotted lines, sometimes for several reasons, you can present defects in these kinetics. What can happen is that then, instead of having a normal contraction that allows the immune system to go back to homeostasis, you may fail the T cells fail to contract, and therefore you will keep having higher numbers that therefore are going to create some issues for the immune system. And in general, we can say that defects in activation of the T cells, proliferation, and apoptosis is going to induce certain pathologies and diseases, such as autoimmunity or like chronic inflammation that then is going to destroy tissues and all that. So as I mentioned before, usually autoimmunity is caused by the dysregulation of many factors, not just one gene or one protein. And there has been multiple genes implicated in several autoimmune diseases, and for example, in systemic lupus erythematosus. So the lab where I started doing my PhD with Dr. Crispin was interested in studying PP2A, that is a phosphatase that has been shown a decade or two ago to be dysregulated in lupus. They were interested in studying the mechanism, how this phosphatase was maybe implicated in the onset of autoimmunity. Something that you need to be aware of is that PP2A is a trimeric enzyme, as you can see here, and it's composed by a catalytic subunit, a scaffold subunit, and as you can see here in yellow, a regulatory subunit. And there's more than 20 regulatory subunits, and each of them really what they do to PP2A is that they will make it like a different phosphatase. So they will present a different substrate and also a different location in the cell. So at the end of the day, each of these trimeric complex is going to be working like a different phosphatase. So that's why it's very important to understand how PP2A is related to these different subunits. So again, the work of my PhD supervisor showed that this subunit B beta, or that is the same as B55 beta, was a regulator for T cell apoptosis, and it was deficient in patients with systemic lupus erythematosus with SLE. So what he found is that if you see here down here, when you activate T cells and then you deprive them of IL-2, they start dying in vitro. And if you quantify this, what he found was that if you compare healthy subjects with patients with SLE, he found two different subsets of patients, ones that were susceptible to death by IL-2 deprivation, and some that were resistant. And further investigation showed that he found that there was a defect in inducing the expression of B beta, B55 beta, in those patients with resistance to apoptosis by cytokine withdrawal. So we were interested to understand how this worked and whether it was something intrinsic to the T cells. So then we decided, okay, let's do a knockout mouse. That's usually a nice approach to study all these mechanisms. And so then we created, this is the gene that encodes for B55 beta, and we created a flux mouse that then we cross into a CD4 Cree, so only the T cells from these mice will be lacking B beta, and the rest of the cells will have the normal dose of B55 beta. However, we didn't find an autoimmune phenotype. So we were a little bit, not upset, but you know, it wasn't what we were expecting. But as I told you before, usually autoimmunity is not just due to one defect. So usually you need to accumulate different anomalies in immune regulation. However, what we found was like with age, these mice accumulate activated central memory CD8 T cells. So we decided, okay, it seems that this defect on B55-beta is causing something to the T cells, so let's go and study it. And I won't mention it for the rest of the talk, but these results have been published recently. So if you have further questions or you want to take a look, that's the reference. So then we set up the question of how does B55-beta regulate T cell immune responses? So for that, we went back to a model of infection that is an intracellular bacteria, and therefore it's contracted for CD8 and type 1 immunity, and this is Listeria monocytogenes. And this is a pathogen that sometimes can be found in contaminated food and can infect humans. In general, with antibiotics, we clear the infection, so it's not life-threatening. But however, in pregnant women, can go to the fetus and cause anomalies in the fetus. So the model we decided, the first approach that we used to study the T cells was infecting the mice with the B55-beta deficient mice with this pathogen, and then analyze the T cell response. As you can see here, at the peak of the response, that is around day 7, when you are starting to have contraction of the T cells, and then at day 30, when you have supposedly again gone back to basal levels of T cells, specific for that pathogen that have generated memory and will be protective against a secondary infection. Yeah, so this is just, so what we did was isolating the T cells, transfer them into congenic mice that we could identify by some markers, and then do an infection of Listeria monocytogenes. And in this case, the Listeria was expressing what is called a model antigen, that is ovalbumin. That is not an antigen, sorry for that. It's not an antigen that is present in the pathogen, but that we can use to study antigen-specific responses because we have a mouse that expresses TCR, a T cell receptor that recognizes ovalbumin. So when we compare knockout and wild-type mice, we were happy to find that indeed there was a difference. And as you can see here, when we were enumerating the number, the numbers of cells at the different days of the response, we found that there was a defect on the T cells that were lacking B55-beta, and though they were still contracting to some extent, this contraction wasn't normal as it happens with wild-type cells. In addition, we found that there was an accumulation, as you can see here on the top, and the quantification in the bottom. There was an accumulation of these T cells that were able to produce interferon gamma, so a pro-inflammatory type 1 cytokine. And again, that the knockouts were accumulating way more of this, so saying that maybe, again, this could be a source of an inflammatory environment. When we analyzed the phenotype of these cells that were not contracting as much as the wild type, what we found was that if we look at effector memory T cells that are a type of T cells, again, all of these are OT1 cells, so they recognize the same antigen in the Listeria, ovalbumin in this case. And what we found was that while there's still a defect in the decrease of effector memory cells identified by these markers, what was striking is that the central memory cells that are supposed to be the ones that eventually will become the source of memory T cells in a secondary infection, if that happens, and can protect us against a secondary infection, they were strikingly not contracting at all, as you can see here in the bottom right corner. So there was, at this point, there was 2 possibilities for this to happening. The most obvious ones, there was maybe there's a defect in apoptosis of the T cells, or maybe there was some anomaly in proliferation. So first, we check the proliferation of these knockout cells, knockout T cells, and as you can see here, when we measure the proliferation of the cells in vitro, the percentage of proliferating cells were quite similar between wild type and knockout. We didn't see any difference. And then we also performed an in vivo experiment to check the proliferation rate of these cells. So as you can see here, we took wild type OT1 cells and conditional knockout OT1 cells with different markers, so we could identify them, transfer them into, again, mice that we could tell which cell was coming from where, and then infect them, and then do an EDU chase experiment where you give EDU to the mice. This EDU is incorporated into proliferating cells, and then you can measure it by flow cytometry. So what we found, and you can see here the different type of cells, like this is the OT1 wild types, the conditional knockout, and these were the endogenous from this mouse. And if you see the measurement of EDU, what we found is that, again, there was both wild type and knockout cells didn't present the same rate of proliferation. Then we went and checked for apoptosis, and again we were happy to find that, as you can see here, the knockout cells presented effect on apoptosis. There was less apoptotic cells as measured by an XIM5 cytops orange staining. This is from the in vivo data, and then on the right you have an in vitro experiment where we also did, when we activate T cells, we anti-CD3, anti-CD28, then put them on IL-2, and then we remove the IL-2 and check how they enter apoptosis. And again, as you can see here, the knockout mice had a defect on this. And the same was true for human T cells. In this case, obviously we couldn't have a knockout human, but we were able to infect the human T cells with a B55-beta short hairpin RNA to downregulate the expression of B55-beta. And as you can see here on the right, also we found that inhibiting B-beta expression decreased the rate of apoptosis in CD8 T cells. I won't go into much detail, but I will show you the data, and then we asked, how was this happening? How was B55-beta controlling the rate of apoptosis in T cells? So we did several screenings in all these molecules that are known to be downstream of TCR and CD28 and promote survival of T cells. And at the end of the day, what we found is that upon IL-2 withdrawal, using this previous method, what we found is that there was, AKT, as Sambi described before, for other things, the activation, the phosphorylation goes away, as you can see here, really soon after removing the IL-2. And if we went again and inhibit B55-beta expression by a short hairpin shRNA, what we found was that normally the T cells will decrease as the phosphorylation of AKT, but if we inhibit B55-beta, this dephosphorylation did not happen as much. The opposite was also true. So if we overexpress B55-beta with lentivirus in human T cells, we found that that by itself will promote apoptosis of the T cells, as shown by an XIM5 staining. And that also, as you can see here, will decrease the phosphorylation, therefore the activation of AKT. We went back to our knockout mice, and again, we found the same thing. Here you have the results from the Western blots, and the quantification here that is easy to see. And as you can see again, like if we compare the fold induction of phosphorylation of AKT on this residue on serine 473, you see that upon IL-2 withdrawal, after 4 hours, you see a decrease in the phosphorylation of AKT. However, the knockout mice do not dephosphorylate as much AKT. Okay, so then it seems quite obvious that maybe B55 is promoting apoptosis by regulating AKT, but how does happen downstream? So one of the ways AKT promotes survival, it's by transcribing, inducing the transcription of survival factors, but also by inhibiting these FOXO factors that, if AKT is activated, phosphorylated in this case, will phosphorylate the FOXO factors, and then they will be sequestered in the cytoplasm. When AKT is not functioning, the FOXO factors are not phosphorylated, and then they can migrate to the nucleus, where they will initiate the transcription of pro-apoptotic factors. So to investigate that possibility, we did an experiment using a plasmid that encodes for FOXO1 with a GFP tag, so we can identify where FOXO was located. And then we infect these cells also with a control plasmid or a B-beta overexpressing plasmid, B55 overexpression plasmid. And what we found, as you can see here, is that, again, with the control plasmid, FOXO was mostly located in the cytoplasm. However, when we overexpress B55 beta, as you can see quite nicely here, the overexpression of B55 beta induced FOXO1 location to the nucleus of the cells. And this also correlated with the induction of certain pro-apoptotic factors like PUMA or HRK. So here we have the link saying that probably the induction of B55 beta will induce the expression of PUMA and HRK, and maybe, not maybe, but this will promote T-cell apoptosis. We went back to the previous result, sorry, was in human cells, or a cell line, as you can on the top. So we went back to our mouse, and then we checked the expression of these pro-apoptotic factors. And in this case, what we found, it was that in the knockout mice, the only difference we found was on this protein called harakiri, HRK, and that as you can see here, the knockout mice, again, fail to upregulate harakiri as much as the wild-type cells. This was an in vitro setting, but the same was true for our in vivo cells from the experiment you saw in the first slides. And as you can see here, HRK on the far left, on the far right, sorry, on day 7 of the infection with Listeria monocytogenes, there was a defect in the levels of HRK compared to wild type. We at that time we couldn't see we didn't have any knockout mice for HRK, but we were interested in studying whether HRK indeed was responsible for inducing apoptosis on T cells. So for that, what we used was, again, we used an shRNA to inhibit the expression of HRK and measure apoptosis in vitro and in vivo. And as you can see, the number of cells when you inhibit HRK decreases, and in vivo, inhibiting HRK expression also to get more activated OT1 cells accumulating in vivo. So therefore saying that HRK seems to be responsible for this. For the sake of time, because it seems I haven't timed myself very well, we tried to identify how B55 beta was being induced. So for that, we focus on the IL-2 receptor and the signaling pathways downstream to try to see whether one of these pathways, the STAT5 pathway, the PI3K, or the MAP kinases pathways were implicated on this. And long story short, what we found was that supplementing by taking out IL-2, but supplementing with IL-7 or IL-15 the T cells, we were able to abrogate the apoptosis of T cells upon IL-2 deprivation. And then we sampled the different signaling pathways with inhibitors, and what we found was only the complete stop or interruption of the IL-2 receptor signaling was able to recapitulate the death of the T cells and the induction of B55-beta. But okay, we came from autoimmunity, so what was the point of all this? Is this defect on B55, is the deficiency of B55-beta responsible or can promote autoimmunity in a permissive environment? So for that, we use again the knockout OT1 cells that we used before, but then we moved to a model where ovalbumin, rather than being expressed on a pathogen, is expressed as a self-antigen. So this mice called Ripple mice. So if there's a breach in tolerance, if the T cells don't control, are not controlled and can attack the self tissues where the antigen is being expressed, in this case, the pancreas and the kidney, then it means that you can develop autoimmunity, diabetes in this case. So we were really happy to see that when we took activated OT1 CD8 cells and transferred them into Ripple mice, these mice, the ones that received the knockout cells, develop diabetes, as you can see here, and only this also happened in the wild type animals and the animals that receive white type cells, but again, lacking B55-beta promoted this. When we went for immunopathology of the pancreas, again, the wild type recipients that didn't express the self-antigen didn't have any obvious inflammation, as you can see here on the top of the immunohistology. However, as you can see here in the islet inflammation score, but especially in the bottom right corner, the mice, the Ripple mice that received the B55 deficient cells really had a lot of damage in the islets and therefore developed diabetes. Finally, we trying to catch up here. Something we observed, if you remember, is that there was an accumulation when we used the listeria model. There was an accumulation of memory T cells, and again, these are responsible for protecting us against secondary responses, infections. So we decided to test whether this extra accumulation of memory cells would be protective. So for this, when we went back, we transferred wild type or knockout OT1 cells, infected the mice with Listeria monocytogenes, and then challenged the mice with a melanoma model that also expressed the ovalbumin, the antigen for the OT1 cells, and analyze what happened. So what we found is that indeed that accumulation of memory cells, when T cells lack B55-beta, was able to increase the survival of the mice challenged with the melanoma. And as you can see here, here you have a PBS control of normal tumor growth, but then those mice receiving OT1 wild type cells were slightly protected against, like, the tumor progression was slowed down, but then the conditional knockouts were significantly more protected, as you can see here, by the tumor growth and the tumor weight. So then, yes, for summarize, this is a little bit the model that we came up that is happening and how B55-beta is controlling T cell activation, rather than T cell activation, T cell apoptosis and the contraction phase of the T cell immune response. So during normal immune response, you have AKT activity and that promotes survival of T cells, and B55 levels are very low. However, because the pathogen has been eliminated and there's no more production of IL-2, for example, IL-2 and other growth factors will go down, and then this will start inducing the expression of B55-beta. And then, therefore, AKT is going to be inhibited by this, and the activity will go down. And that will induce the appearance of FOXO. FOXO will be responsible for transcribing, in this case at least, HRK, and that will bring apoptosis to the cells and the contraction phase of the T cells. However, as putting it back to the perspective of autoimmunity, what we show is that then if you have an abnormal induction of B55-beta, this could contribute to autoimmunity onset. Again, by itself may not be sufficient to drive autoimmunity, but definitely will contribute to the appearance of autoimmunity. And indeed, we also published last year this paper where we found that in autoimmune diseases, there's an hypermethylation and a reduction of the expression of B55-beta that is responsible for a defect in apoptosis on T cells. And with that, I really want to thank all my present lab, the Mackenzie lab here at Cambridge, but also the Crispin Rossetti lab, where I did my PhD and a small postdoc, and where we did all the work that you guys have seen. And again, this especially thanks to my supervisor, because this is the project he started as a research associate, and then I took and finished with his help and the help of all the people of the lab and all the funders. And thank you guys for listening to it. Thanks a lot.
Thanks so much for that. That was a really great talk. So we have one question from an anonymous attendee asking, is B55B expression restricted to T cells, or is it also expressed in other immune cells?
So indeed, B55, how it was shown before to be expressed, is expressed a lot in neurons in the brain, and the effects on the regulation of the expressions causes other diseases. But in the immune system, it's expressed in other cells. I think it's expressed also on B cells and dendritic cells, and indeed, as far as we know. We are taking a look, well, no, I already move on working here, but my PhD lab is working on studying the function of this in other immune cells.
Cool. We'll take one more question, just in the interest of time. So someone else is asking, does B55B knockout, or do B55B knockout T cells, are they less prone to apoptosis in other conditions like strong TCR signaling or TGF-beta signaling? And is STAT5 a repressor of B55B?
So we didn't check all the conditions, but some data with fast and fast ligand experiments, and also the patients from the samples from lupus patients didn't show any defect on fast and fast ligand signaling. And so the T cells were still prone to apoptosis through those mechanisms. However, again, we cannot discard other ways like a strong TCR signaling. We didn't check that. And as far as I remember, nor TGF-beta. There's no difference on Tregs in these mice or in this immune response. So, but again, we don't know if there's something else going on there. And about the STAT5, we really tried, we thought and we were hoping for STAT5 to be the main driver, or like the repressor, as you said, of B55-beta. But the experiments we did trying to do overexpression, but also using inhibitors of STAT5 didn't induce the expression of B55-beta. So far we don't have, we think it's the contribution of all the signaling pathways downstream of the IL-2 receptor, rather than just the STAT5 or ERK or some other. Thanks.
That's cool. Thank you so much. It was a really great talk.
Thanks a lot to all of you guys.
So we will move on now to Marlene.
Hello.
So hello, Marlene.
Yeah, can you hear me?
I can
see me. Cool.
And we can see you as well. So Marlene is a postdoc at the University of Edinburgh, which is where we met. and she did her PhD with Cecile Benezet, where I also did a rotation project, and Marlene supervised me. And then she went on to work with, was it Mary Britton?
Yeah.
Yep. Before moving into Rebecca Gentech. Is it Gentech? Is that how you pronounce the surname?
Yeah, I guess so. I don't know. She's German. Ask her.
Where she is now, and she's going to talk to us about time for a lipid break, or Tim for a lipid break, while what your Tim for macrophages eat. So thank you very much, and take it away.
Yeah, thank you very much, Matt, for the introduction, and thanks for the invite. Obviously, you're doing a great job with the STEM Village, so thank you. So today I'm going to present you my PhD project, actually. So this is a study I did with Cecile back in the days. No, it wasn't too long ago. So like Matt said, it's a time for Team 4 a lipid break. So this was a pun because I was scared that if I was showing you the real title of the talk, nobody would come, because it's heavily adipose and fat heavy. So the real title of the talk, and also the title of the manuscript that is actually in revision, that Cecile is revising with Pete Smith, thanks for that as well. The actual title and the full title is Regulation of ABCA1 plus resident adipose tissue macrophages and postprandial cholesterol levels by Team 4. So it's a bit of a long title. I'm going to break it down for you. You have three components in this title. I'm going to talk about resident adipose tissue macrophages. I'm going to talk about postprandial cholesterol, so cholesterol that you get after a high-fat meal, and also Team 4. I will explain later on what is Team 4, basically. So first of all, adipose tissue macrophages. So they are obviously very abundant in adipose tissue, and they adapt and contribute to adipose tissue function, but also dysfunction. So they are in both types of adipose tissue. So in a lean individual, in the adipose tissue of a normal or lean individual, you have mostly type 2 immune cells and also mostly M2 macrophages. But in the opposite of the spectrum, during obesity, adipose tissue macrophages adipose tissue accumulates macrophages, and these ones are bad for you. They are M1-like macrophages. They are inflammatory. So this paper, for example, showed that they express CD8 very highly, they're inflammatory. There's a switch between M2, M1 macrophage during obesity, but I never really liked this M1 or M2 macrophage denomination, because for me, macrophages in tissues, they are very heterogeneous. You have plenty of markers. And up until recently, actually, the definition of macrophage in adipose tissue was very limited. And as I just finished my PhD like 2 years ago, 1 year ago, there was 2 papers that came out that described a bit more adipose tissue macrophages. So for example, in this paper, they described the vasculature-associated adipose tissue macrophages, or VAM. So these ones are beneficial. They promote glucose homeostasis, for example. And this paper, also in 2019, talked about lipid buffering macrophages that are heavily recruited during obesity and actually are beneficial for the tissue. And these macrophages express TREM2. So try to remember this macrophage, because I'm going to refer to them a bit later on. So this was just a brief introduction about macrophages. And now let's have a brief introduction about fat. So I'm sure you're a good person, and Monday, Tuesday, Wednesday, Thursday, you're having a healthy meal, lots of greens, 5 a day, you're perfect, because maybe you don't have a long experiment or flow plan, so you have time to do this. But maybe on the Friday, maybe you finish at 9 p.m., the flow wasn't working, your qPCR wasn't working, so you go to the chippy. And on the chippy, you're gonna get what? A butter sausage, and also all of these kind of things that you can find in Scotland. But what happens when you eat fat? What happens in your body? So it's going to be very simplistic, and maybe I'm going to miss some paths, but I think I will try to keep it simple for you to understand and not be scared, because maybe for some of you, it reminds them of a nightmare they have during uni, all of these lipids, metabolism, pathways and stuff. So when you eat fat, so it arrives in your gut, and your gut are going to pack the fat into kilomicrons. So the kilomicrons are high in triglycerides. So they're going to go in your bloodstream after a while, and they're going to go into your muscle, they're going to go into your fats, and they're going to be lysed. They're going to be lysed by lipoprotein lipase. So the lipoprotein lipase are going to break down the lipids, and some of the lipids or free fatty acid are going to be taken up by your adipose tissue to be stored once again. So once this breakdown has been done, you're going to have what's left. So it's a kilomicron remnants. These kilomicron remnants are going to go in the liver. In the liver is going to do its magics once again. It's going to repack some of the free fatty acids and combine them into VLDL. The VLDL are going to go all the way around into your muscle, into your adipose tissue, and they're going to be transformed, sort of speaking, into LDL. So LDL contain a lot of cholesterol, and maybe if you look at a blood analysis at the GP or whatever, you will remember that LDL are bad for you. They are the bad cholesterols. And these LDLs are actually the cause of atherosclerosis. So atherosclerosis is when you have the buildup of fat within your vessel. But you also have macrophages within your vessels. And actually, during atherosclerosis, some studies have shown that macrophages can do what's called a reverse transport. So they can transform lipids into cholesterol and eject them back via ABCA1. So ABCA1 is a transporter of cholesterol, a cholesterol efflux transporter. So by doing this, it's avoiding the accumulation of lipids in the macrophage and avoid the macrophage to be dysfunctional. So you eject this cholesterol, and this cholesterol is going to be back in the circulation and pick up by HDL. So now if you remember also your GP and the analysis, HDL is a good, it is the good cholesterol, it's good for you because this HDL, contrary to the LDL, can be excreted via the bile. So it's good, you don't have it anymore. So now I'm talking to you about fat, I'm going to you about macrophages, but what's the link? So actually, in human, in genome-wide association studies, several papers, like 5, 6, 7 papers, have associated TIMD4 with dyslipidemia. So they associated this gene with altered level of LDL, altered level of cholesterol, or triglycerides in the plasma. So TIMD4, you might recognize this gene because actually TIM4 is highly expressed on macrophages. Also TIM4, so, you know, I did a PhD about TIM4 and I wrote a thesis about TIM4, but still I have to Google every time what TIM4 stands for. TIM4 stands for T-cell immunoglobulin and mucin domain containing 4. More importantly, TIM4 is a phosphatidylserine receptor. So TIM4 is going to recognize phosphatidylserine at the surface of apoptotic cells. So this is their eat me signal. And by recognizing this phosphatidylserine, TIM4 is going to engulf the apoptotic cells to protect you from, dysfunction or etc. So TIM4 is expressed on a lot of resident macrophages in the body. For example, it's expressed on liver macrophages. They also know as Kupffer cells. They're expressed in the gut, in the peritoneal cavity, in the heart, in tumor-associated macrophages. So they are very abundant. So combining these two observations, TIM4 is associated with dyslipidemia, TIM4 is on macrophages in the body. So can TIM4 actually impact lipid metabolism the same way as some macrophages during atherosclerosis? So just an overview of what I'm going to talk about just after. I'm going to talk about the adipose tissue macrophages heterogeneity in lean mice, their origin, and then I'm going to talk about the impact of 1 day of high-fat diets on this adipose tissue macrophage, the role of TIM4, and then conclude with a proposed mechanism. So I think you must be used by now to look at this data. So if you want to show heterogeneity, you're going to be using single-cell RNA-seq. That's how people now define heterogeneity. So what we did is we took the adipose tissue from lean mice, either lean mice after a controlled diet, so a normal diet, or lean mice after a one night, so a one meal of high-fat diet. Then we sorted these macrophages and we gated these macrophages as being F480 positive cells. So I'm first going to start with the clustering, and then I'm going to refer a bit later on about the impact of the one-day fat diets. So regarding the clustering, we could define, or actually Jordan Portman, he did all the analysis. Thank you, Jordan. So he could see 8 clusters. So in these 8 clusters, you can see here, you can see some of the genes that are expressed in these different clusters. Actually, we could define 2 groups. Like the clusters on the right here were expressing highly ADGRE1. So it's basically the gene coding for F480. So they were F480 high. And they were resembling really like resident macrophages in other tissues. So they were expressing live1, they were expressing F480 high, a low CCR2, for example, and also TIM4. So here you can find a cluster, the TIMD4 cluster. And here on the left, you could see the CCR2 positive more macrophages. So they were expressing high level of CCR2, low level of live1, and then high level of other genes that are actually expressed in monocyte-derived macrophages. So here you could also find the LAM, so the lipid-associated macrophages that usually you find in obese adipose tissue. So here just saying that in the lean, you can also find this kind of macrophages. And here as a little remark is that actually some DCs were in the set. So here we just marked them and they were the cluster number 7. So here, I guess you can just appreciate that the adipose tissue compartment, macrophages compartment is highly heterogeneous in lean mice. And also you have this dichotomy of maybe some resident macrophages and some macrophages that resemble a bit more the monocyte, the freshly monocyte-derived macrophages. So next, we just checked by flow all of these markers because it's very important to see what they express in reality. So this is the gating strategy I used, and the gating strategy I'm going to refer to through all of the talk. So here, so LOX is low, F480, and then in the F480 population, we can look at F480 high macrophages. In this F480 high, you could see the TIM4 positive macrophages and also TIM4 negative macrophages, and then you could see the F480 low macrophages. So here you can see a bit of the expression. So actually the cluster 1, the blue cluster is here because it's F480 high, TIM4 positive, live1 positive. So grossly, like we could find all of the markers, all of these clusters by flow. So that was good. But are macrophages recruited or resident? Because I just talked a bit about maybe the origin, but I haven't made any experiments to look at it. So here is the experiment we made to look at their origins. So to look at their origins, we did what's called the tissue-protected bone marrow chimeras. So I'm going to try to explain it very simply, and in the hope that for those that are not very maybe aware of this technique would understand. So we protected the adipose tissue with a shield, and then we irradiated the back legs of the mice. And then the next day, we injected the donor bone marrow. So the donor bone marrow is going to be either from a wild type or from a CCR2 knockout bone marrow. And after 8 weeks, we looked at the chimerism. So the chimerism, so the higher the chimerism is going to be, the higher the population is going to be bone marrow dependent. CCR2 is basically a negative control because the CCR2 is essential for the recruitment of bone marrow-derived monocytes. So that means that if a population have a low chimerism in this context, that means they are really bone marrow dependent. So here is the flow of what we can look at. So like I mentioned, in the tissue, the monocytes, they had a high chimerism of 26%, and it was CCR2 dependent. As you can see here, there's barely any cells coming from the CCR2 knockout bone marrow. The eosinophils, they had a high chimerism because they're bone marrow derived. Actually, they're not CCR2 dependent, which is also logical. And if you look at the macrophages, so when I say macrophages, I'm talking about F480 CD8 B cell. You can see they have a lower chimerism compared to the monocytes in the tissue, but it was also CCR2 dependent. But if you break down the macrophage population into the subset, into the TIM4 positive, TIM4 negative, F480 low population, you can actually see that there is actually an heterogeneity in the bone marrow dependency. So the F480 low had a high bone marrow dependency, TIM4 negative had the intermediate bone marrow dependency, and the TIM4 had a very low, the lowest bone marrow dependency. So they were the true residents of the adipose tissue. So it's just a bit confirming what we saw by single cell. In the interest of time, I might skip a bit this, but actually we looked at the impacts of obesity on this turnover. So we did exactly the same kind of experiment, except we put the mice on the high-fat diet for 8 weeks, so a long high-fat diet, so they become very fat, and then compare it to the control diet. And the main, the conclusion of this experiment very quickly is that the high-fat diet did not change the bone marrow dependency for TIM4. So actually the TIM4 macrophage during high-fat diets, even if the dogma is saying that macrophages are recruited during high-fat diets, actually the TIM4, they were proliferating. If you have more questions about this, I can answer it after, but just I'm trying to keep it short. So let's go back one step. So now I'm just going to talk about the single cell data, and I'm going to talk a bit more about the difference between the clusters in terms of genetic expression and in terms of their maybe function. So here you have the resident macrophages that are TIM4 live-1 positive, and here you have the more monocytic derived macrophages, which was the cluster 4. So what are their genetic differences? So actually, if you do a pathway analysis and you compare the genes here on the right compared to the other genes expressed by these other macrophages, you can see that they have a high endocytic capacity and lysosomal content. So they express genes linked with lysosome, with endocytosis, with arteriosclerosis, with phagosome. And when you look at it by flow cytometry, actually these TIM4 positive macrophages, they had a lot of lysosomes, and their lysosome were very active, as seen with lysotracker staining, and they also contain a lot of lipid droplets or just lipids actually. So here is just the graph showing exactly the same thing as the flow. So compared to the two other populations, TIM4 had the highest lipid content, the highest number of lysosomes, and the highest lysosomal activity. So when you look at this data, it just indicates that macrophages in the adipose tissue, they might be able to uptake more lipids and metabolize them a bit more because they contain lysosome and they can do lysosomal hydrolysis. So let's just look quickly at some pictures because it's always very pretty to look at pictures of these macrophages. So here is the adipose tissue, you have adipocytes, and here squeezed against the adipose tissue, you have these macrophages. So in gray here, you can see TIM4. In red, you can see the lysotracker. And here, if you zoom in, you can see in green the lipids. So actually, sometimes we could see that or even most of the times, we can see that the lysosome, TIM4 and lipids were forming like they were colocalizing a lot. So we started to ask ourselves, oh, is TIM4 actually involved in lipid uptake? And is it involved in lipid uptake in a way that's going to bring it to the lysosome for lipid digestion? So more pictures, and also pictures of the human adipose tissue, because human, they actually have a lot of TIM4 macrophages in their adipose tissue, and they are full of lipids, full of lysosome, and they all colocalize. So this looks good for our hypothesis, actually. So if we compare the control diet and the high-fat diet by single cell, and we compare like all of the clusters, the cluster 1, so the resident adipose tissue macrophages, they undergo a lot of transcription. They have a transcriptional activation that is barely seen, for example, for the cluster 5 and 4. So you can see a bit in cluster 3, but really the cluster 1 is the one that is the most changed by the 1 day of high-fat diet. And you can see here that some genes linked with the response to lipid and the regulation of plasma lipoprotein particles levels are activated. So one of these genes, if you remember what I said at the beginning, is ABCA1, and ABCA1 is linked with the reverse cholesterol transport. So it's just expulsing fat from macrophages. So to avoid any lipotoxicity, for example, or maybe to recycle lipids, and maybe so the lipids can be uptaken by HDL, for example. So here you can see the cluster 1 between the control diet and the high-fat diet. They highly express ABCA1 and even overexpress ABCA1 during high-fat diets. So here is just to show that even at steady states, macrophages that are TIM4 positive, they have a lot of ABCA1 at their surface compared to the other macrophages. So just to summarize a tiny bit, because then after we're going to jump to mechanistical experiments. What I just showed with the single cell and the flow is that TIM4 macrophages, they have a lot of lysosomes, they have a lot of lipids, they express ABCA1. With high-fat diet, they increase the lipid and the lysosomal pathways, and they increase ABCA1 expression. But okay, but what's the role of TIM4? Because TIM4 is a phosphatidylserine receptor. We need to know what's the actual role of this receptor, because even though these macrophages seems to react to lipids, maybe to engulf them or digest them, we still don't know what TIM4 is for. So what we did, we used antibodies and we injected mice with TIM4 antibodies. So to block TIM4, to block the surface of these macrophages. Because the lysosome seems so important in the way the TIM4 macrophages worked, we also use chloroquine. So it was way before chloroquine became very cool or uncool for COVID. But actually the purpose of chloroquine is to increase the lysosomal pH and inhibits lysosomal hydrolysis. So basically they inhibit the lysosomal activity. So what we did is put control diets, mice on control diets, and put mice over 1 day of high-fat diet, and we blocked TIM4, or we injected the control, or we injected chloroquine, and then we looked at the lipid levels. So 1 night of high-fat diet increase free fatty acids, increase cholesterol, increase HDL cholesterol, and the non-HDL cholesterol. But remarkably, if you block TIM4 or if you use chloroquine to block the lysosomes, you're not going to see the increase of cholesterol, or you're not going to see the increase in HDL cholesterol. And if you look at the lysosomal activity, by blocking TIM4, you will also block the lysosomal activity that is induced usually by high-fat diets. And this will be the same with chloroquine, because chloroquine blocks lysosomal activity. So TIM4 seems very important for the lysosomal activity of these macrophages, and also to maybe release the HDL cholesterol and to participate in metabolism of lipids. So because I don't know if I have time, I'm going to skip this one. But basically, we found that because TIM4 is expressed everywhere, actually the liver and the PEC, or the peritoneal exudate cells, are not involved in this mechanism. So maybe it's really the macrophages expressed on the adipose tissue that are producing the HDL cholesterol. So this is just a summary to finish in the proposed model. So after a meal, like I said earlier, your fat is going into your adipose tissue. And in the adipose tissue, you're going to have these TIM4 macrophages. These TIM4 macrophages are going to be able to capture the fat, bring it to the lysosome, and digest the lipids via the lysosome. And then once the digestion is complete, they're going to release cholesterol into the bloodstream. And the cholesterol can be packed into the HDL and just excreted out. So that's it for me. So I think, I would like to thank everybody. So in Cecile's lab, Pete, obviously, that is keep on working and keep on doing the revision for the paper, and Lucy and everybody else. And thank you for your attention.
Thank you so much, Marlene. That was really, really cool.
Sorry, I was trying to be quick because I saw like it's 5 and I'm like, oh no, it's 5. I need to finish.
No, don't worry, don't worry. So Cecile has a question. So if you give me 1 second, Cecile, and I will wait. Okay, Cecile, you should be able to talk, I think.
Sorry, I didn't have a question. I don't know why.
She just wanted to say hi. I was like, you have a question, but she...
Sorry about that.
Well, while we wait for anyone to write any questions in the Q&A box. So wait, let's see. So Noe said, amazing talk. Fred Sheedy has asked, said great work, and asked, is there any evidence for lysosomal dysfunction in TIM4 positive ATMs?
I would say no. Like I would say TIM4 macrophage is actually very active and very lysosomal functional. I don't know if, for example, TIM4 deficient macrophages or in TIM4 knockout macrophages, if you're going to see any things like this. So I would say just no evidence.
One question I had was, obviously, it seems really clear the role of TIM4 in macrophages, but does it have a similar function, or is it known if it has a similar function in T cells, since it's called T cell?
No, it's just TIM4. So TIM4 can interact with the TIM1, and TIM1 is on T cells, and it's going to help them proliferate. So I haven't looked at the impact of the TIM4 blockade in T cell, biology. Mm-hmm. But that's something that could be interesting. It's just because, with the high-fat diet, the one day high-fat diet, I doubt that there is such a response in the adipose tissue. But it would be interesting to look at the interaction between macrophages and T cells. Yeah,
absolutely. Can add that to the list of things to do. So if no one, oh no, we have another question. Okay, so thanks for the great talk. I'd like to know if you've looked at this a little longer term, and if there's any relationship to the development of insulin resistance. That's the first part, and then I'll come on to the next part.
Yeah, so I'm reading the question. So yeah, we tried to do a blockade. So I actually did GTTs. So I did a long alpha diet, and I blocked TIM4, but because it's IP injections, after a while, and the mice are becoming so fat that when I did the flow at the end of the experiment, I could see some TIM4 in the adipose tissue, so the blocking wasn't great. But when I did the GTT, so to measure the glucose tolerance, there was one experiment showed that there was a difference, that the mice injected with the TIM4 were a tiny bit more insulin not resistant, sorry, the opposite, sensitive, sorry. So the mice injected with TIM4 were insulin sensitive compared to the other mice. But then I did the repeat experiment, and I show less of an impact. But because I wasn't really blocking efficiently the macrophages, we didn't know how to actually, conclude on this experiment. And then the second question is about CD36, because obviously TIM4 is a phosphatidylserine receptor, and so it's phosphatidylserine. You have some in your lipids, but what's going to pick up the most the lipids in macrophages is CD36. There is no doubt about this. CD36 is the main lipid uptaker. But I'm just wondering, and what one of my hypotheses is that TIM4 can maybe form a complex with CD36 maybe to pick up lipids that would maybe express a bit more phosphatidylserine at the surface. I don't expect TIM4 to be alone in this process, but I suspect that TIM4 positive macrophages are going to be better at processing the fat. Mitochondrial respiration, and I have no idea, but I'm very interested into this. So I don't know what they do with the fat, if they can use it for OXPHOS or not.
Yeah, it'd be really cool to do some seahorse or Ouroboros.
Yes, but these macrophages are very hard to culture in plates.
That's
They lose TIM4 expression, so they're very hard.
No, that's really, really cool. If anyone else has any questions, can they contact you both? And if so, would you just put your Twitter and email in the chat box, please? That would be great. And then I'll just give people a second to copy it rather than logging straight out.
There. Sorry, I don't remember my email address.
Cool. Well, thank you very much, everybody, for coming along. And thank you for those both fantastic talks. Next week we have, is it next week that we have it? Yes, next week we've got a triple bill. So next week we have 3 talks coming up, starting at 4 o'clock, which I will be shouting out about on Twitter. So please come and join us again. And I look forward to seeing you all next week. Bye, everyone.